Now we'll move on to our next speaker, who is Dr. Jennifer Collinger from the University of Pittsburgh. Dr. Collinger is a professor in the Department of Physical Medicine and Rehabilitation at the University of Pittsburgh, Rehab Neural Engineering Labs with secondary appointments in the Department of Bioengineering at the University of Pittsburgh and the Department of Biomedical Engineering at Carnegie Mellon University. She is a past president of the International Brain-Computer Interface Society, and her research interests are related to the use of neuroprosthetics to restore function for individuals with upper limb paralysis or loss. And yeah, we all look forward to your talk. Thank you so much.
Thank you for having me here today. I think this is the middle of my presentation, not the beginning. Yeah, thank you. So I'm going to talk about some of our research in the lab trying to develop bidirectional brain-computer interfaces. I'll focus on that bidirectional aspect for the first half of the talk, and then I'll move into talking about what we're really trying to do in order to control hand function. So a bidirectional BCI that we are developing in our lab uses electrodes that are implanted in the brain. We record from motor cortex and try to decode the patterns of activity and turn that into a control signal for a device such as a robotic arm. We can also record from sensors in the robotic arm and turn that into stimulation patterns that we can play through electrodes that are implanted in somatosensory cortex to restore the sense of touch. Oops. So far in our clinical trial, we have had 6 study participants. They're split across the University of Pittsburgh, where I work, as well as the second site at the University of Chicago. Most of the participants have had spinal cord injury resulting in loss of hand function or upper limb paralysis. And these studies have been conducted under FDA investigational device exemptions. You can see a picture of the device that we actually use in the lower right-hand corner. It's an intracortical electrode array made by Blackrock Neurotech. In order to calibrate a brain-computer interface, we need to understand how brain activity is related to movement. And so typically people have imagine or attempt to perform different actions while we record neural activity. And so here you're seeing an example of a BCI calibration paradigm that's for reach and grasp control. They're thinking about moving this virtual robotic arm to a point in space indicated here by this red object. They're instructed to grasp, maybe carry it to a new location and orientation, and then to release. And so we can know the kinematics is being controlled by the computer. We can record the brain activity and figure out that relationship. And so now many decades of research have shown that attempted movements generate similar neural activity to that measured during overt movements. And so we can actually explicitly model what we expect this neural activity to look like and what that relationship to kinematics is. And I'll show you a little bit of data, but it's been known now for more than a decade that people with spinal cord injury can use these neural population recordings to enable simultaneous and continuous control of endpoint translation, orientation, and grasp. And here's an example of what this looks like. This is our very first participant. She's controlling the robotic arm with 7 degrees of freedom, so 3D translation velocity, 3-dimensional orientation velocity, and grasp. And so the computer is not performing any of this. This is all completely under her control. Her task was to just place this object on the shelf and put it back down. And you can see she's thinking about the trajectory of that action. She's not thinking about forward, down, left, right. It's really a natural and intuitive control signal. And so this really took what had been done in basic science non-human primate labs and said, okay, we can actually do this in somebody with paralysis, chronic paralysis. And we really wanted to move on quickly to trying to restore natural sensory motor function and adding in the capability to provide sensory feedback. So why do we need sensory feedback? That control looked pretty decent, maybe not as fast as an able-bodied person, but it was a good start. Well, certainly as we move towards more dexterous activities like buttoning a button, tying a shoe, brushing your teeth, you're really going to rely on that tactile feedback in order to do that fine motor control. We still don't necessarily know how to enable motor control of those actions, and so even for simple things like maintaining a stable grasp, handing an object to another person, holding onto a fragile object, that's all primarily mediated by the sense of touch, not vision, even when you're able to see your action. And so that was our motivation for trying to add sensory feedback. And I told you that we implanted electrodes in somatosensory cortex, but we need to know where to implant them. And so this is work we've been developing over each of the implants, trying to improve our imaging procedures. And we published last year the results of these procedures for our first 5 participants who had sensory arrays. This work was led by John Downey, Feng Liu, and Hunter Schoen. So each of the 5 participants underwent pre-surgical imaging, either with fMRI or MEG. And the task was to attempt to move each of their digits many times in the instructed sequence. And so here I'm showing you fMRI data from one of the participants, C1. We're zooming in on motor cortex, it's indicated there by M1, and somatosensory cortex, indicated by S1. And that's exactly what we're trying to map and try to decide where to place these electrodes. And so this participant had a really sort of beautiful map of their finger activity. The colors correspond to the digits as you see on the left-hand side. So red is thumb. That's most lateral, as you would expect. Then moving to index, middle, ring, and pinky is kind of small there. It's primarily located in the sulcus in those recordings, but you can see where we would expect that to be. And this map looks a lot like an able-bodied person. So these representations seem to be preserved after chronic spinal cord injury. So we use this imaging to decide where we're going to implant these electrodes. But then the next question is, okay, did that actually work? Were we able to generate sensations in those areas? And so we came up with a procedure. It's pretty basic, but where we essentially map what the participant is feeling through each electrode. And I should mention that the sensory feedback work at the University of Pittsburgh is led by Rob Gaunt. His postdoc, Ceci Verberschat, collected and analyzed a lot of this data. The sensory feedback work at University of Chicago and for the team was originally led by Suleiman Benzmaya, who unfortunately passed away 2 years ago, and it's now being led by Charles Greenspan. But the procedures that they came up with were to, every month or so, stimulate the electrodes in a random order. If the participant felt something, they told us, and then we tried to characterize that stimulation. You can see our stimulation parameters here. We sort of settled on these as the standard parameters that we use for each of these mapping surveys. If they feel something, they tell us where on the hand do they feel it, how natural is it, what's the modality, does it feel like pressure, does it feel like tingling, and we try to characterize that and look at the stability over time. So now I'll show you just results from 2 of the participants into how well this pre-surgical mapping actually corresponded to the sensations that we could generate with intracortical microstimulation. So this is the same fMRI image that we looked at a couple of slides ago from participant C1. The 2 checkerboard-like patterns are actually sort of a blow-up of what the 2 somatosensory electrode arrays look like. So every other electrode is electrically connected. That's why it looks like a checkerboard pattern. The color of each of those squares corresponds to when you stimulate that electrode, where did they report feeling it? So if you start at the bottom, you see a couple of electrodes are colored red. That means the participant felt it on their thumb, and that lines up well with the imaging data that we had. As you move more medially or up on this page, you see that it's yellow, and then it moves into green and blue on the upper electrode. And so this corresponded really well with the functional imaging that we had done prior to implant. For participant P2, who is actually the first participant to get sensory electrodes in our study, we had used MEG to map electrodes. We only mapped 3 of the digits, so the thumb, index, and pinky finger. You can see, though, we still see that same preserved organization with the thumb most lateral, followed by index, and pinky as most medial. And so he had had his electrodes implanted for about 8 years, so the sensations shift very slightly over time, and he tended to have sensations that were more at the bottom of his fingers, maybe spanning multiple digits. But you still see this gradient where all of the red activity and yellow activity is located more on the lateral array, the bottom one here in this image, and then the middle ring and pinky is located on the medial array, as would be expected. So just briefly, I'll show you exactly where our participants have felt these sensations. So these maps are showing overall time for however many surveys we had done with each of these participants, what kind of coverage of the hand did we get. So this is putting all of the electrodes together. So for participant P2, his sensations were all localized to the hand, mostly at the base of the digits, and we could talk a little bit about why that is in the Q&A. For participant P3, we had broader coverage of the digits, mostly from the thumb to pinky. P4, we only had one survey at the time this paper came out. He had coverage mostly of the last 3 digits in the palm. Participant C1 had very focal coverage of his fingertips. Hopefully you can see it in here, of the first 3 fingers. And participant C2 had a very similar pattern of coverage of the fingertips. So this is good. In people with chronic spinal cord injury, we're able to localize the somatosensory cortex. Their hand representation seems to be preserved, and we can generate sensations that feel like they're coming from their own hand. In order to be useful, we also need to be able to convey the amount of force that is being felt by the robotic arm, for example. So in this, we use a magnitude estimation task. This is showing data from a single electrode and single participant, where we deliver stimulation with amplitudes in a random order and then ask them to report the magnitude of that. And you can see that, for this electrode, it's a very linear mapping between stimulation amplitude on the x-axis and reported magnitude on the y-axis. And just last year, we published a paper with data from 3 participants and multiple electrodes. The different participants are shown here as different colors. And you can see that as stimulation amplitude increased, the reported intensity also increased in a fairly linear manner. So this is useful to us as we're thinking about applying sensory feedback in a real-world case. And a couple of years ago, we actually did this. We put this together for a bidirectional BCI. So this schematic shows the major components, similar to the very first slide that I showed you. But this participant was controlling a robotic arm using activity from their motor cortex with 5 degrees of freedom, 3D translation, pronation and supination of the wrist, and then opening and closing the hand. At the same time, they were receiving sensory feedback. So we were recording from torque sensors in the motors, and we mapped that torque onto electrodes corresponding to each of those digits with a linear encoding function. And this was a big undertaking led by Charlene Flesher and John Downey, who were grad students in the lab at the time. Jeff Weiss was an engineer who kind of made this whole system work and developed artifact rejection techniques that were really key to making this work. And I've mentioned Rob Gaunt already as leading the sensory feedback efforts in our lab. So one of the ways that we quantified performance was on a test called the Action Research Arm Test. It's a common clinical outcome measure that assesses unilateral arm function after stroke. We adapted it a little bit for our brain-computer interface study, but essentially it's grabbing objects of different sizes and shapes, picking them up and moving them to a new location. So it assesses your ability to coordinate arm and hand actions. We scored each of those different objects on a scale from 0 to 3, where 3 means that you could pick it up and move it to the new location in under 5 seconds, which is considered able-bodied time. We did 9 different objects, so the maximum score here is 27. A score of 2 means that you successfully completed it, but it took you longer than that 5 seconds, but still less than 2 minutes. My clicker, okay. Okay, so prior to actually doing kind of a controlled experiment where we had that torque sensor mapping like I described with multiple channels of sensory feedback, he had performed this task over a couple of years, and his median score on this task was 18. There were a couple of sessions where he had single channel sensory feedback, but we were sort of changing the different settings. Then we came in and changed nothing else except to fix the ICMS parameters, the sensory feedback parameters to that mapping that I showed you, and the score increased from 18 to 21. In the next 4 sessions, we took that sensory feedback away, still had vision, and his score dropped back down to his original level, to a score of about 17 this time. I think these scores, you're like, okay, it's statistically significant. What does this actually look like? I'll show you this video, which is a side-by-side comparison of performance. So on the left, you're going to see the best trial with sensory feedback, and on the right, the best trial without sensory feedback. So still vision, but not tactile feedback. And what you'll notice is on the left, every time they move very directly towards the target, and as soon as they touch it, they have confidence that they have the object in their hands and can pick it up and move it over. So we'll watch this one again. On the left, moving directly towards the object, picks it up. On the right, even though they can see it, not very confident that the fingers in the back have actually a hold of that ball, and so it takes them longer to pick it up and move it over. And all of the different objects are the same. I guess I'll let this video play out now that we're watching it. So if we quantify this over those sort of 4 days of fixed performance without ICMS, the average completion time for the different objects was about 21 seconds when they were only using vision. When we added in sensory feedback for those 4 sessions, the completion time on average was cut in half. So this was a dramatic improvement. I think the score doesn't quite convey how fast, but I think you can see in the video and in this completion time that it was really, quite a bit faster. And there's more detailed analysis in the paper, but it really came from that time after object contact when they were receiving sensory feedback. They were just much more confident that they had the object in their grasp and could pick it up and complete that action. So for the last part of my talk, I'm going to talk about what we've been doing lately to move towards more dexterous control. So the ability to individuate digits or control the amount of force that a person is applying through their hand, because that's really what we envision as being possible when you have that sensory feedback component. So in order to get at this, Brian DeCleva, who is a research scientist in my lab, he designed this task, which is pretty straightforward in its design. It was essentially instructing people to think about pressing each of their 5 digits. So you can see the 5 different buttons corresponding to 5 fingers. They're cued which button to press with that white circle. And as soon as they press that button with their brain activity, it turns into a color, and then they're instructed to release. And so we calibrated this very similarly to how I showed you that virtual environment before, where it's initially under computer control and they just think about that action. We recorded their neural activity from about 200 channels that were implanted in motor cortex to figure out that relationship between this instructed finger press and neural activity. So the first thing that Brian did was just plot the trial averaged sort of firing rate estimates for each of the channels. And what you can see here is you're already starting to see a pattern where a lot of the neurons have kind of similar responses at the beginning and end of that action. So he performed factor analysis to transform those 200 channels to about 20 factors and plotted them averaged over which finger was being pressed. So here are the responses when they're pressing the thumb. And we can fill in, here are the responses when they were instructed to press each of these digits. And what you can start to appreciate even without any additional processing is that some of these components look the same for each of the digits, and some of them are very different. And so Brian wanted to try to leverage this to better understand, okay, why are there some that look the same and why are there some that are different, and can we leverage that to improve control? So he first looked at the components that were the same. And what he found that these tended to convey sort of temporally specific events in the action. So one that he called the onset component was active sort of at the initiation of that press and was similar for all 5 digits. There was also one that was active during the hold phase. So for as long as that button was pressed, we could see activity along this dimension in the neural space. And then there was also an offset component that was active at release. So if we line all of those up, and then look at, okay, but when is the finger-specific information most robustly represented in the population? You can see that it lags that sort of population, that common onset component by a little bit. It's about 200 to 250 milliseconds, depending on the participant. And what we mean by finger specificity is if you do an analysis, such as linear discriminant analysis, that we can maximally separate the neural activity patterns that are associated with each of these individual digits. So using this information, Brian came up with something that he called the gated decoder design. It has 4 components. The first is that we detect the onset. So for any type of finger pressing action where you see this really robust onset transient. So we can detect that pretty robustly. Then once you're in that sort of finger-specific phase, you can actually try and look and classify which finger they are trying to press. We can define a threshold of confidence that we want to have. As soon as we're confident that we know what finger they want to press, we can initiate that click. And then they hold on to that click until we detect the offset transient. And so in this way, they don't have to think about continuously pressing that finger. We detect what it is, we hold it, and then they release using that sort of non-specific release component. You can see what I'll play in this video. On the left is actually online BCI control performance that our participant is thinking about pressing each of these actions. Again, the cue is the white circle. As soon as it turns colored, it means that they're pressing it. On the right, I'll show you the neural state. And so first, we're going to look at how that neural activity in a low-dimensional space kind of bounces around between these finger-specific representations. And hopefully it'll play. OK. So on the left, you can see they're pretty quick at being able to actually press these fingers when they're cued. Again, it was about 200 to 250 millisecond latency on our ability to decode this. And you can see that the neural activity between each trial kind of goes to the center and then moves out to this finger-specific representation in neural space. If you rotate that activity around, you can actually see that there's an orthogonal dimension that is indicating the onset and offset of that click. So all of the finger colors are now clustered together, and there's this really large amount of variance in the neural activity associated with the onset and offset of that action. And so that's what we rely on for our decoding. We quantified performance in the two different participants. So this is the participant whose video I just showed you. He had both of his electrode arrays implanted in the hand-specific region of motor cortex. And so this is just a confusion matrix. So what was the target finger on the top, the decoded finger on the bottom, and it's a really nice diagonal. The accuracies ranged from 94 to 100% depending on the digits across multiple sessions and about 500 trials. Participant P2, you see still a diagonal pattern. There tends to be some confusion between adjacent digits. His success rates range between 42 and 87%, depending on the digit for this task, again, across multiple trials and multiple days and about 600 trials. He had been implanted for about 8 years whenever we ran this experiment, and so anybody who's worked with intercortical electrodes know that signal quality does tend to decline over time. He also had one electrode array in hand area and one in more shoulder arm reaching area. And so we were really relying on information from one array for this performance. So it still was a good indication that this may be a viable approach. Well, can't get it to click forward. OK. So we use the same idea to look at how grasp force might be encoded. So Brian asked the participants to think about grasping an object with a gentle or firm force. And what you can see in the plot right below it is in blue. In two participants, there's one solid line, one dashed line. We see, again, a robust signal signaling the onset of that grasp. And then in red, a robust signal at the end of that release. On the bottom, what I'm showing you is how much variance is associated with the amount of grasp force that they are trying to use. And you can see that it peaks. I don't have a pointer, but it peaks kind of right after that initial onset transient and then decreases over time. It's even more dramatic when we ask them to hold an object with a low or high force and then think about carrying it to a new location. So again, you see that onset component in blue, the release component in red, and this force-specific variance, again, it peaks right after that initial onset, but then it decays and is really absent during that translation and release phase. So across a number of experiments, we saw that this grasp force information decreased over time, and particularly during concurrent arm movements. And so the idea of trying to decode this, this variable force continuously did not seem to be a viable approach. We tried it. It doesn't work in the same way that you can decode translation. So we've started using this gated decoding approach as a generalized method for being able to provide control. So we're always reading in neural activity. There's nothing stopping us from having one pathway that's decoding continuous action. So for translation and reaching, we see that information represented continuously. We can continue to do that. But for hand-specific variables, now we're trying to gate it using these onset and offset transients that could be used for finger decoding, which digit you're pressing, for grasp force decoding, and we're even using this for other hand shapes now. So very briefly, I'll just show you what this might look like. And as an example, on the bottom left, you can see there's target forces shown in red, and the decoded force now is shown here in black. And so what we're really doing is detecting when do they want to start and stop applying force, in that initial period classifying what force we think they are trying to apply, and then we hold it. So the force traces are very clean, but they are generally lining up well with the target force, which is what that scatter plot is showing. On the right-hand side, the task was just to grab an object with a low or high force and then carry it back and forth across the table as many times as you can in about a minute. And so again, you can see they're able to apply either a low or high force and maintain that while they continue to perform the translation action. And just wrapping up, we use this for multi-click, so left and right mouse click. Here the participant is controlling 2D translation velocity, and we can decode the left or right click, which is indicated by the color of the sad face that needs to be rescued. And so they're able to do this pretty robustly. And this method is just much less susceptible to like early drops when you're trying to click and drag. And this was really important for our participants who use the system at home for doing things like creating art and doing digital painting. So I'm just going to end with 2 slides showing you, I know this conference is about neuroscience in the everyday world. This is what our lab looks like. And we certainly do a lot of our experiments actually in the participant's home, but it's kind of just taking this lab and putting it in their living room. So it is in their home. It's not quite realistic, but we have made some progress in this area, and I know you'll hear, I'm sure later in the neurotechnology panel, there are now companies that are actually doing clinical trials in the home with wireless devices, and that's amazing. We did our first version of the portable system in 2019. Jeff Weiss actually had a paper. I realized I forgot citations on this page, but I'm happy to share it with you. In 2019 with a portable system showing that it provided comparable performance to what we had in the lab. And then in 2021, Brian DeCleva published a paper using data that had been collected entirely independently by our participant at home. We had given it to him in January of 2020. And as you know, the labs shut down, and so we were able to work with him virtually to conduct experiments during COVID. So this is the second generation of the in-home system that you can see pictured here. Participants use it for things like creating art or playing games, and we now have students who are looking at learning and sort of the impact of changes in mental state in home environments. So it was a little bit of a whirlwind tour of some of the research that's going on in our lab, but I showed you at the very beginning that BCIs can enable high degree of freedom robotic arm control in people with chronic paralysis, that we can use intracortical microstimulation of somatosensory cortex to restore tactile percepts in somebody with chronic spinal cord injury, and that this can improve BCI-controlled grasping. And then in the second half of the talk, I showed you that motor cortex carries information that is specific about different digits or different forces, as well as that is non-specific, just about the timing of grasping-related actions. We can harness that with a gated decoding approach to deliver much more stable and robust control in a variety of tasks and even in different domains that our participants are using at home. So with that, I'd like to thank my lab and, of course, the study participants who devote a ton of time to coming into the lab to do this research. We couldn't do it without them. Thank you.
Thank you so much. That was very exciting. Do we have a few immediate questions for Dr. Collinger? I do have a question, maybe until the others kind of can think of something. So I feel like bringing this type of technology to the actual like real world everyday life, one question that always pops up for me is like, do you think, or do you know, like, what's the individual variability of people responding to this? Also, like, for example, will everybody be able to learn controlling this type of device, or is there something like some people might not actually show a response and then they have implanted electrodes, but it doesn't work as well, or does it depend on the stage of the disease when it's implanted? Do you know, like, how is the variability of the response?
Yeah, we're starting to get a sense just from the field as a whole. But what I would say is if you are getting good recordings, right? So right now these devices are still investigational devices. And so it's a tedious surgery. And to be able to put them in, I'm sure you're going to hear later from Dan and Erling, who's going to talk about how to solve this problem. But yeah, if you're getting good recordings, BCI control tends to come very quickly for people, so it's not something that needs to be learned when you're using these implanted electrodes. We're picking up on natural sort of motor plans that are preserved. I think it's different if you're talking about neurodegenerative conditions such as ALS, because cells in motor cortex actually die and there's atrophy, and so it's a little bit different there. But I think for people with spinal cord injury, if you're getting good recordings, you're going to get good control. Depending where you place them, you could get better, better or worse if you're targeting the right brain area in motor cortex.
Thank you.
Thank you so much for your talk. It was very cool. I was wondering, for the arm reach task, were you able to look at how the responses changed over time as they learned to better use the device? You mentioned that with the sensory feedback, participants are more confident that they have control over the device. So I'd imagine for both the sensory and the non-sensory, as they gain confidence using the device, that the responsive time might be variable.
Yeah, we've looked for evidence of learning quite extensively, and we don't see much, surprisingly. So even looking from the day that we changed to giving sensory feedback, did we see some learning even over the course of those first couple trials? We really didn't, and it puzzles us a little bit because most motor tasks, right, you do get better over time. I think there's a couple potential reasons. One is that we're sampling from so few neurons, and those might even change from day to day. And so potentially they just don't really have that closed loop feedback and in sufficient magnitude to drive that learning. It may also be that we don't give them enough learning pressure to really try to improve. And so we see that performance can be variable, but we don't see a steady trend in learning, surprisingly.
Very interesting. Thank you.
Mm-hmm.
We have a Zoom question. It will probably be a long time before we know the answer to this, but it seems reasonable that a younger person would learn to do these tasks faster than adults, just as younger people learn complex video games faster and are generally faster at them. Do you agree?
Yeah. Well, so I think what we have picked up on so far is really these natural motor plans that have evolved over time. And so all of the participants in our study were able to move their arm at some point and then had a traumatic injury that resulted in paralysis. So I think they had kind of already gone through that learning and development stage. As we think about sort of pushing the bounds of this, if you wanted to map this onto a video game controller, for example, or control something that was not an arm or that they never had an arm, I do agree, like generally people who are younger are more likely to be able to learn those more arbitrary patterns.
Hi, thank you for your talk. It's really exciting work. Earlier you mentioned about the neurons for the sensory touch will shift over time. So I'm just wondering, will the same thing happen with the control neurons? And if you're even able to recalibrate the stimulation or the decoding mechanism, like say 5 years after they get the implant?
Yeah. So on the control side, things change rapidly. They can change even in the course of hours or minutes even, and so that's much more of a problem. On the sensory feedback side, it actually tends to be much more stable. So I did say that they change over time. It's on much slower timescales, and I think has to do more with either the arrays potentially settling into cortex and allowing us to target a new layer, or perhaps the sensation is a little bit weak and they're getting maybe some natural sensation from their own hand and they need to learn to recognize it. But for the most part, when you stimulate an electrode, it's going to feel like it's in that exact same area for time courses of months to years. So those maps tend to be fairly rigid. I think in the paper that Charles published last year, they showed the average centroid of the projected field only changed by about 2 millimeters over the course of 2 to 8 years. So it's pretty small.
All right, thank you.
Mm-hmm.
Thank you for the wonderful talk. It may be too soon to say. I'm kind of thinking that the people who you induct into your studies probably are on medications. They hurt themselves. They're on anxiolytics. They're on pain medications. They're on muscle relaxants. Maybe they're on stimulants. Have you noticed any trends toward medications that either improve response or make responses worse?
I think if they're on muscle relaxants or things that cause fatigue, we do tend to notice a decrease in performance. It's pretty hard to say because we don't necessarily track that very robustly, and they're on those medications chronically. And so to some level, they kind of have their own individual baseline that we're working with. They tend to come into our lab at the same time every day, and so we catch them at the same time in their medication cycle. But I agree that for real-world use, probably the amount of engagement and attention that they have is going to change, and we definitely see correlates of that induced by other factors. So whether it's fatigue or distraction or something, as that sort of level of engagement changes, it does change the motor activity.
You have to make sure they got a good night of sleep, or is there some period during the day where they're optimally trained?
Most of our participants report poor sleep chronically. And so they don't need to be awake. I think certainly peak performance would likely come if people were well rested and alert, but it's not the case that we need to catch them during their sort of magical hour. They're able to use this device pretty robustly all the time.
Thank you.
Mm-hmm.
All right, thank you so much. Let's thank our speaker again. The next step would be that I'd like to ask all speakers of the symposium to come back on stage, and we have our general Q&A. And at the same time, I encourage everybody to formulate your questions and come to the microphone here and ask questions to all the speakers that might be relevant, maybe for one or two or to everyone. That would be great.
Okay, hello, this is Hasan Ayaz from Drexel University, and I have a question for Dr. Maren Bixen. Well, thank you for sharing your impressive solution for the scalability challenge of neurotechnologies, and you addressed both cost, miniaturization, and user interface simplification. So do you think one of these dimensions is more important than the other? And if you further go deep down in one, you can improve the scalability potential. Like, for example, if you further miniaturize the system, would it help? It looks like you still have potential on all these. And the follow-up on that is, can you comment on the regulatory pathway that you picked for this? Because that is the efficacy demonstration of the overall system. Thank you.
It's a big question. I'll give you a quick answer. We're not worried about miniaturization right now. I mean, the active elements are in the milligram range. And so actually the size is almost all the enclosure. So that's one of all the problems we've had. That's not one of the problems you've had. I mean, the other issue, you really talk about things like cost and scalability. And I think this is really the difference between, let's say, academic-centered research and really thinking about translation. If you're at that laboratory scale, it's okay to make 100 a month, or maybe okay to make 1,000 a month. In this case, scalability means, making 1 million a month. You know what I mean? And it's just a completely different scale of process. And same thing with cost. Again, if you're satisfied staying at that academic stage, frankly, you can make each one for $100, right? As long as the grants allow it. While when you're thinking about translation, obviously, the pressure becomes very, very high to get to sort of a Band-Aid or a pill level. So there's no simple answer to that other than to say that, really with the translation, the pressures are high. And as far as regulatory, as long as we can keep this in the non-significant risk range, we can make that process much simpler. And I didn't talk about it, but a lot of the engineering went into risk management. These things are, I say borderline edible, and then someone says eat it. But they certainly won't, the electrochemistry prevents an excessive discharge.
While others think about their questions, I have another question actually I think it applies pretty much to all of you. Like, when I think about bringing this, from the lab into the real world, I think what's interesting to me is also like the long-term effects, of stimulation or BCI usage. And I find it hard to distinguish, like, for example, things like gait in the course of disease probably like get worse anyways, right? So how can you distinguish like things if the kind of the treatment effect decreases over time? Do you know? So A, the question is like, do you know if that happens? Does the effect decrease over time? And how do you know if that's because of the disease getting worse, or is that something that is more like maybe technology, like, I don't know, impedances getting worse, or the system kind of habituates or adapts or something like that?
I can take a stab. So for the gait-related research, these patients have been implanted for over 2-3 years now. And actually, Ken did analysis in looking at stability of the biomarkers over that time span. And for most of the subjects, it has been surprisingly stable over a year or so in terms of the biomarker from when he first tried to whittle it down versus the latest visit. So I think, I guess maybe that's advantage of less invasive in terms of like non-intracortical, right? With ECOG and local field potential, the signal seems to be pretty stable. But you're right, in terms of disease course, with disease progression, like their dynamics are going to change. Yeah, we haven't found a great way of addressing it besides, periodically checking in to see how well these, classifiers are performing.
Yeah, for brain-computer interfaces, for intracortical electrodes, until the last year or two, all of the human work had used that device that I showed you, the Utah array. And so it's a rigid device. It's mechanically mismatched from the brain, and the materials tend to degrade over time. So I think that's every device that's implanted, the signal quality does degrade, and then therefore performance is lost. There are now other devices that are actually in clinical trials led by companies that are using flexible materials or targeting different structures, like even trying to use intravascular approaches. All of these have trade-offs. I think it depends, like what level of information do you actually need to get from the brain? If you just need an on-off signal, you probably don't need an intracortical electrode. If you need that level of movement, though, there needs to be advances in the materials and design in order to preserve that longevity. And just, as the other speakers were saying, as for people who have neurodegenerative conditions, certainly you're fighting an additional factor where the actual information that you're able to extract does degrade over time as well.
That was a long one. Hi, mine's quite a high-level question, but when we think about scalability and neurotechnology, how do we think about how we engage people with this technology? So we kind of tell people, we're going to do this kind of work with your brain, you're implanting deep brain stimulation, for example, and we're thinking about trying to do this over millions of people. How do we engage? For example, I work with a lab in Sheffield who do DBS, and they've had neurologists that have never seen someone from a BME group, minoritized ethnic group. How do we engage people like that in this technology? How can we work with different groups to make them understand what we're doing and how we can integrate it wider into society, and how we're doing that moving forward?
Yeah, I mean, I do think it's a big challenge. Right? I was really struck in your talk of there've been therapies that have existed that people just don't adopt, right? Because the actual process of getting them is too challenging. And so that's certainly where we are with brain-computer interfaces. Only a handful of surgeons in the world know how to implant these devices right now. It takes a team of people to calibrate them and to keep them operational. Even in our case, when we do have a home system that people can use, some participants have caregivers who are really willing to learn how to attach the device and kind of get things set up from day to day, and others don't. And so I know that a lot of groups and companies are working to develop systems that are kind of always on, that the person themselves can use, that requires wireless technology that does exist now. But I think that there's still going to be some hesitancy, right? The benefit needs to outweigh kind of the risks and challenges associated with doing that in order for people to start to adopt it. I would say that implanted technology does have lots of applications, just deep brain stimulation, even pacemakers for a heart. Sometimes you do need to implant in the body to deliver the amount of function that you are promising, right? A wearable would be great for a BCI, but we don't have access to the level of movement-related information that we need to restore upper limb function, to restore speech, and so you do need an implanted technology, but it needs to be, something that can be scaled. The surgical approaches need to change, the cost needs to be lower, and it needs to work all of the time. Reliability and accuracy requirements for assistive technology is very high, and so we have a pretty high bar that we need to go after.
Yeah, and just from like the movement disorder perspective, DBS is one of the most underpenetrated and underutilized technology. There's so much class 1 evidence that it works great, yet only probably 10 to 20% of the patients who qualify actually end up getting it. But I think this is a power, in terms of scalability, like a power of industry collaboration, right? Medtronic now, their state-of-the-art generator that everyone's putting in now has this sensing function, and you can activate it. And adaptive DBS, a few months ago was FDA approved. So this is possible. I think as it generates more interest, and there are a lot of patients who advocate and who want it, hopefully that will spread this knowledge and interest beyond just a few academic centers that are really invested in the technology and understanding it. And now with a lot of the data that the companies have access to, like NeuroPace and Medtronic, it would be a fantastic industry, academia, research collaborative to understand what the signals are doing on a much larger, like hundreds of patients, hundreds of thousands of patient scale.
It's a hard question what you asked. I think it's sort of what maybe partially what this conference is about. There's this question of how much do we need to do educationally, how much do we need to do versus advocacy, or is the tech just not good enough? You know what I mean? And so it goes back to the engineers and the people who are pioneering translation. And maybe the second part of it is true as well. DBS has been around for a long time, but maybe it's been begging for adaptive technology, and that's what it's needed. And same thing, with prosthesis, the fact that we need, to integrate these other harder technologies, these sensory things. So sort of blame the technology, not just the education. The both are clearly important.
All right. Another question that I had on my mind, it's probably, again, relevant probably to all of you, but I certainly saw it very clearly in Dr. Bixen's talk. Like, you need to think probably a lot about factors that go beyond just research, right? Thinking about like bringing this technology to like actual people, or to the clinic, or to everyday life. So, and again, that's probably a huge question, but like, could you say just a few words about like what are the actual like the biggest challenges that you're facing, like bringing this technology, these approaches from the laboratory to the actual like people to use it, or clinicians to use it, or people at home?
I think one thing, and I think this is true for all of us, is we need to recognize, you have the sort of the academic front, and then there's industry. And I don't think anything that ultimately, when you get to patients, ultimately labs don't build this technology. We like to think, okay, we're academics, we do everything, but we don't build medical devices. We're not qualified, regulatory-wise qualified to bring medical devices. So that's one thing. I think it's you have your industry here. I know these other speakers must work very closely with the companies that they deploy. Maybe that's something to think in mind about how to be more open to those academic industry collaborations, and figure out how to do that ultimately, to the best benefit for patients.
Yeah, I completely agree with that. On the device side, that's not going to be my role of trying to improve the device and making a device that gets implanted perfectly every time and gives you the best signal quality. But I think what we've seen, kind of as I've already said, is the reliability requirements are high. And so a lot of what we're trying to do in the lab is try to push the science, try to understand behavior, see how that's impacted by sensory feedback. But the first products need to be something that works every single time in every single situation, no matter if the person is sleepy or on a different medication or in a different environment. And so I don't think that first generation of products is going to have the degree of function that you see from different academic research labs. but it's going to be based on those scientific findings and collaborating with industry to deliver something that is really robust and performs well.
Yeah, and the scalability is a huge question, and I think like Medtronic is running into that now with the deployment of adaptive. Like, how do we actually use it in patients? And even a center like as accomplished and well experienced as UCSF, even our group is having challenges in like who's the best candidate? How do we track this? And it involves a lot of patient engagement. It's not like, you know, we implant the device, we turn it on, it's like a light switch. It takes, you know, we're distilling like months and years of data intense collection with like postdoc students analyzing it to figure out what works for that patient. But if you're trying to do it on a mass scale, that's really challenging. So, and then Medtronic, you know, they have to now hire, you know, PhD neuroscientists to kind of figure out that problem as well. So yeah, I think it goes both sides.
Okay, perfect. Thank you so much for all your answers, for all the wonderful insights. I think with that we're gonna.
Sorry, can I?
Yeah. Do you have another question? Yeah, please. Go ahead.
Okay. Yeah, hi. I'm Melanie from the University of Oldenburg. Yeah, my question is mainly directed towards Dr. Wang, but maybe Dr. Collinger and Brixen, you also have something to add. So I was very intrigued by the swimming study because I also did EEG during swimming, so yeah, that was something nice to see. And yeah, so of course I'm also interested in swimming, and so I was wondering whether it might be a therapy approach as well to like take out the gravity from walking and to like either walking in the water environment or also just swimming to try to improve like motor function in patients with movement disorders, because I think there are also studies reporting that some Parkinson patients can like cycle when they're like on a spinning bike. Yeah, if we take those motor function that are still somehow still present in those patients and try to train them, that maybe functions like walking might also be improved over time?
Yeah, that's definitely a really interesting idea. I think I think there are physical therapy rehabilitation programs using like aquatherapy for patients not only with Parkinson's but with like injury and neurological diseases in terms of motor function recovery. And I think one of the things, actually I trained a physical therapist in my lab who completed a PhD. It just, you know, we do all these therapies and there's no neurological basis. So the effects are often short-lived and it's not targeted. So I think one of the main motivation is like if we can understand what specifically within the brain circuit is disabled and whether we can train that circuit to prevent freezing, preventing falls, that would be a lot more fruitful, I think, to couple like rehabilitation with an understanding of targeted neuroscience approach.
Thank you again. All right, I think with that we're gonna finish that session. Thanks all our speakers again very much.